Good morning all of you. Welcome in my channel PharmaZonner. I am Gaurav Sharma. Today we will discuss about cleaning validation questions. Which one is asking during the interview. So let's start.
Number 49. Cleaning validation demonstrates or cleaning validation is the procedure to ensure that cleaning process adequately and consistently removes product residues, process residues, and environmental contaminants from the cleaned equipment or system. Next important question: when we should perform cleaning validation? Answer of this question is cleaning validation should be conducted for the initial qualification of manufacturing process or equipment. Further, periodically, cleaning verifications shall be done. In case of major change in the cleaning procedure or equipment trend, we should perform cleaning validation. Next, very frequently asked question: which guidelines are referred for cleaning validation? Here, 6 majorly referred guidelines are mentioned. You can pause this presentation here and note it down. First one is US FDA's guidance for cleaning processes. Second, EMA's guidance for cleaning validation. Third, WHO's guidance on cleaning validation. Fourth, PICS guideline, and fifth and sixth are PDS technical report number 29 and 49. Next important question: what is term MACO, commonly called MACO, NOEL, and PDE used in cleaning validation? Answer is MACO is maximum allowable carryover. NOEL is amount of a drug in mg that does not have any effect on the human health, and PDE is permitted daily exposure of the active in the clean product in micrograms, that is mcg. Next, very frequently asked question: what is formula for MACO calculation? You can see a formula for MACO calculation on the screen. It is a multiplication of 3 factors. First one is TD, which is a single therapeutic dose. Second is BS, that is the batch size of the next product to be manufactured in the same equipment, and third factor is the safety factor. This multiplication is then divided by the LDD value, which is the largest daily dose of the next product to be manufactured in the same equipment. Next, very frequently asked question is why 3 cleaning cycles are considered during cleaning validation run? Answer of this question is consecutive 3 cleaning cycles data gives enough statistical information to conclude on the repeatability of results. 1 or 2 batches data may found insufficient, and obtaining data from more batches probably will give repetitive results. Additionally, PDA, that is an International Association for Pharmaceutical Science and Technology, in technical report number 29 suggests to take 3 consecutive cleaning cycles run during cleaning validation. Next important question: what is dirty hole time? The dirty hole time is a cleaning process challenge that is defined as the time from the end of manufacture until the beginning of the wet cleaning process. This dirty hole time shall be specified in cleaning validation protocol of the product or group of products. Next important question: what is clean hole time? The clean hole time is defined as the time from the end of the validated cleaning process until the beginning of use of that equipment for manufacture of product. This clean hole time may be performed as a part of the cleaning process validation protocol or may be performed as a standalone protocol. Next important question: which hold times shall be validated during cleaning validation? As per PDA, the maximum allowable hold time for a piece of equipment shall be validated for following 3 cases. First case, after use before cleaning, which is dirty hold. Second is after cleaning before reuse or re-cleaning, which is a clean hold. And third case is, if appropriate, after cleaning before sanitation should be validated. What are the commonly used sampling techniques during cleaning validation? Answer is rinse sampling and swab or wipe sampling are the most commonly used sampling techniques during cleaning validation. Along with these 2 techniques, other techniques used are coupon sampling, solvent sampling, placebo sampling, and product sampling. Next question: what you should do first, rinse or swab if you are doing both? Answer is it is advisable to perform final rinse before swab sampling, so no interference from the swab sampling could affect the rinse sample. Next important question: what are the advantages and limitations of rinse sampling? There are many advantages of rinse sampling, out of which 3 are mentioned here. First one is this technique is very easy to sample. Secondly, this is a less technique dependent than swab sampling, and third, it allows sampling of large surface areas. It has some disadvantages, like in some cases it lowers the test sensitivity. Inability to detect location of residues is it's one of the disadvantage. And third, rinse volume is critical to ensure accurate interpretation of results. Next question, what are the basic requirements for rinse sampling? Basic requirements for rinse sampling are: rinse solvent should dissolve the target residue. Second requirement is rinse solvent should reach all product contact surface areas. And third requirement is surfaces should be rinsed to ensure complete coverage of equipment surface and sufficient removal of target residues. Next question, what are the advantages and limitations of swab sampling? Three major advantages of swab sampling are: first, it dissolves and physically removes sample. Second, this technique is adaptable to wide variety of surfaces. And third, this technique is economical and widely available. Whereas it has some disadvantages, like this technique may introduce fibers into the sample. Second, this is technique dependent and results may vary based on the technique applied. Third disadvantage is swab material and design may inhibit recovery and specificity of the method. And fourth disadvantage is evaluation of large, complex and hard to reach areas is difficult with swab sampling technique. Important question, which key parameters shall be considered for preparation of risk assessment for cleaning validation? Answer is, below are the 5 key parameters that needs to be considered for risk assessment. First one is identification of the possible location of residue deposition. Second, sequence of cleaning procedure to be followed. Third, important steps which needs supervision. Fourth, type of sampling aids. And fifth one is the selection of tool required to clean. Next question is, what is equipment grouping and product grouping? In equipment grouping, identical or similar equipment can be grouped. Equipment grouping can be done through scientific rationale that equipment having same design and construction can be grouped for validation purpose. This may reduce the total number of validation runs necessary to demonstrate consistency of cleaning process. And in case of product grouping, products may be grouped if they are cleaned by the same cleaning process. Product grouping can be done considering the solubility, ADE or PD levels and cleanability. Next question, what are the CIP systems? CIP systems are clean in place systems, and this refers to an automated system which can be a single pass or a recirculatory system. CIP system generally utilizes a spraying device to provide coverage and physical spraying on equipment surface. Examples are manufacturing tanks, blenders, fluid bed dryers, reactors and fermentation tanks. Next question, which study shall be performed for cleaning agents during cleaning validation? Answer is, during the cleaning validation cycle, the quantities of cleaning agents, their concentration and their addition rate must be studied. Methods for storage, expiration dating, inventory control shall be covered during cleaning validation. Next question, why TOC testing is done during cleaning validation? Answer of this question is, the total organic carbon or TOC test is fast and effective analytical technique to evaluate the cleaning of manufacturing equipment. This technique can help to ensure that the cleaning processes meet the predetermined acceptance criteria. Increase the presence of total organic carbon in sample directly indicates presence of large molecular weight compounds, that is inadequate cleaning. Next question, what are the non-specific analytical tests for cleaning verification? There are 5 majorly used non-specific analytical tests for cleaning verification. These are TOC, that is total organic carbon, second pH, third titration, fourth conductivity, and fifth is gravimetric analysis. Last and important question, how we can enhance training practices of cleaning procedure. Here are the 5 tools or the techniques given with which we can enhance training practices of cleaning procedure. First, we can use checklist to determine that all operations are carried out in a proper sequence and documented. Secondly, we can do periodic monitoring of cleaning procedure. Third, we can use dedicated or assigned cleaning personnel. Fourth, we can take feedback from operators to modify procedures. And last one, we can use video to demonstrate proper cleaning operation technique.